So the way that three-dimensional structure occurs within proteins is limited because of the peptide bond's ability to rotate. This is because of resonance, but generally you can think about the rotation around the bonds of a amino acid and a peptide as being described as two different types of angles. We call this the phi angle and the psi angle. Essentially, this is the angle of the carbon amide nitrogen bond, which I'll show you in a second. This is the phi angle. And the psi angle is the carbon-carbonyl angle bond. If this was a straight peptide, these angles would be 180 degrees. That is the angle degree you would see. And the organization around the peptide bond paired with the R groups is what is going to determine the structure, the secondary structure of a peptide. So these angles, the flipping of these angles and movement of these angles will determine the secondary structure. And again, that's this portion shown here, which we'll get into more detail here. Again, secondary structure is determined based on these phi and psi angles around the peptide bond. So here's what that looks like. I know it's hard to just describe without seeing it. Here is one carbon. This is the alpha carbon. This is the central carbon of an amino acid, and it's in a peptide, right? It's in a chain. It's connected to a bunch of other. You can see it has an amine attachment here, and it has a carbonyl attachment here. And here is its R group. So those are the three distinguishing factors of this amino acid within the peptide bond. So the angle of this central carbonyl to its nitrogen is one of those angles. And the angle that consists between this carbon and the carbonyl bond is the other angle. So again, you can think about these as essentially rotating planes, as you see here. And depending on their orientation, you can get different secondary structure. But it's very limited. They can only orient in so many ways. I mean, again, if you think about your two hands flattened out, flat oriented, you can only put your hands in certain conformations, right? I can't twist them and curl them. So there's only so many ways a peptide bond can angle itself, but depending on those angles, depending on the movement of those bonds, you will get different secondary structure. So some of these angles, some of the combinations of the phi and psi combinations are unfavorable, and that's because of what we call steric crowding. Too many large, bulky molecules near each other. They're crowding the backbone of the atoms and the side chains. And some of the combinations are very favorable because they allow for more hydrogen bonding interactions to occur. So this is key. This is why we end up having only a subset of secondary structures, because most of those combinations end up just not being favorable for the structure of the molecule, of the peptide. And so we end up with secondary structures that are very, very classified, and you see them in protein from one to the other. The two most common arrangements of secondary structure that you will see are an alpha helix and a beta sheet. And again, this is because the movement of those peptide bonds, these two types of structures are the most energetically favorable of those rotations. The alpha helix is stabilized by hydrogen bonds between nearby amino acids, or residues. We'll use this word residues to describe an amino acid within a peptide chain. Beta sheets are also stabilized by hydrogen bonds. So here's the key thing, both of them stabilized by hydrogen bonds. These hydrogen bonds are between adjacent segments. So the amino acids might be very, very far away from each other if you were to look at the primary code, just the sequence. They might actually be amino acid number 5 and amino acid number 100, but they're stabilized by these hydrogen bonds that hold together those strands. Anytime you have any irregular arrangement of a peptide chain, we call this a random coil. So again, we're going to be focusing on secondary structures that are the alpha helix and beta sheet, but you can have other types of secondary structures called random coils that don't fit these exact specifications. So we can actually determine an alpha helix and a beta sheet numerically based on those angles. So they're characterized based on the phi and psi angles, the actual positioning of the peptide bond. That angle is what characterizes something as having an alpha helix secondary structure or a beta sheet. And within the beta sheet, there's actually subtypes of beta sheets, which we'll break down as well. So let's go more into the alpha helix. Again, this is one of the main types of secondary structures. And again, it is held together by hydrogen bonding. This is a helical structure, a helix structure held by hydrogen bonds between the amides of the amino acid. So not the R groups, not the special R groups, the amide portion, the NH portion of amino acids of an N and N plus 4 amino acids. That means that every fourth amino acid is making a hydrogen bond with the first amino acid. So N and N plus 4 amino acid is going to have a hydrogen bond between it using the amide. Okay, it's a right-handed helix, which we'll discuss more. Its direction of turn is right-handed with 3.6 residues per turn. Every single one loop of that turn has about 3.6 amino acids in it. So almost 4 amino acids. And that makes sense based on what I just said, because we have our N and N plus 4 amino acids that are kind of lining up and making a bond. The peptide bonds are parallel with the helix. So the peptide bond, the actual bond holding together the amino acids, kind of runs the shape of the helix. Whereas the side chains, those R groups of the amino acid, you can see they stick out. They're perpendicular to the helical structure. So they stick out of the helix, whereas the actual peptide bond is kind of flat and straight with the helix as it turns. So what is a right-handed helix? Again, this has to do with the rotation of the turn in the helical structure. And to remember which is right-handed and which is left-handed, you can use your actual hands. So a right-handed helix, if you were to point up and look at the way that your fingers curl, that is the same direction of rotation and curl that a right-handed helix would have. Whereas if you were to look at a left-handed helix, similarly, if you point your thumb up in the air, the way that your fingers curl is the same way that the loops of the helix loop. More about the alpha helix. So let's look at the alpha helix kind of from a top-down way, because I think this helps you envision where the bonds are happening. So this is like if we were to look at the inside of the alpha helix, top down, looking down through, and you'll see that the inner diameter, this diameter is very small. It's actually so small that just generally nothing, or it's too small to fit anything inside. So the R groups cannot fit inside the helix because that diameter is small. And that again is why they end up pointing outward to the outside of the helix. So these purple groups are the special R groups, whatever they may be of that particular amino acid. Now, the outer diameter of the helix is about double the size. So we went from something 4 to 5 angstroms in size, that's the inside diameter, and the outside is about 10 to 12 angstroms. And it just so happens that this size, 10 to 12 angstroms, also happens to fit very well inside the groove, the major groove of the helix of DNA. So these alpha helixes tend to be associated with areas that help to bind onto DNA. Now you can see the numbering of amino acids as we go along the coil. So here is amino acid 1, 2, 3, and you can see amino acid 4. So amino acid 1 and amino acid 4 are the ones that are forming a hydrogen bond using the amine group and the amine nitrogen and the oxygen of the carbonyl of the opposite pair. So here we have this plus representing the positive charged polarity side of a hydrogen bond and the negative charged polarity side of a hydrogen bond. Remember that hydrogen bonds form off of elements, normally nitrogen and oxygen, and they form between the hydrogen of one element and the lone pairs of another. In this case, we're using this NH portion. This is the amide portion of one amino acid and the carbonyl oxygen of another amino acid, and that's where the bond is forming. So this amino acid number 1 is represented with a slightly positive charge, meaning it's using its amide. The hydrogen is slightly positive, and the carbonyl on amino acid number 4 is its lone pairs, which are slightly negative, are making the hydrogen bond with amino acid number 1. And you can see this then continues as it goes around. So there are hydrogen bonds that are lining all the way around down the alpha helix, holding it together. Another key thing is that amino acid 1 and number 4, even though they are hydrogen bonding, they're not actually aligned directly on top of each other. They're a little bit askew as shown in this cartoon. Now, amino acid number 1 and number 8, so the 8th amino acid away, are actually aligned directly on top of each other. So you can see every 8 amino acids, you have a essentially perfect alignment of the coil. And again, that just keeps repeating as you go down. So a question here is based on looking at this kind of structure and where the amino acids are as you go through the alpha helix, if you wanted to have an alpha helix with a hydrophobic side, essentially, of one part of it is hydrophobic and the other part of it is not, where would you put your nonpolar amino acids? Well, you can see based on the way that these alpha helixes coil, you can have every second and third amino acid and then 6th amino acid. If it were to be a non-polar side chain, then that whole side of the coil, as it kept going, if every second, third, and 6th amino acid had a non-polar R group, then that whole side of the coil would end up having a hydrophobic side. a hydrophobic face, whereas the other side of the coil would not necessarily be nonpolar. So not all polypeptides are going to have a alpha helix structure in them. Um, and the sequence of the polypeptides, which actual amino acids are in there, will affect if something will have an alpha helix or not. So some small hydrophobic amino acids like alanine and leucine actually are strong helix formers. So they tend to make it more likely for an alpha helix to form. Proline actually acts as an alpha helix breaker, and that's because the way that proline forms, or proline is a special amino acid that actually kind of binds on itself. This breaks the rotation ability of an alpha helix, and so the bond of an alpha helix becomes impossible if a proline is added in. So prolines will completely break up an alpha helix. Glycine also acts as a helix breaker. You would think at first it wouldn't because it is a small residue, just like alanine and leucine, but it's so small, the tiny R group actually ends up supporting other conformations that help with bending. And in this case, that actually breaks the helix. So glycine and proline both are helix breakers. Any attractive or repulsive interactions that would occur between the side chains of a 3 to 4 amino acids will also affect the formation. So if those R groups within the structure of the alpha helix have repulsive or attractive interactions, it could either help stabilize or it would help break an alpha helix. And again, that's just going to be depending on if they are attractive or repulsive. So here's actually a chart that shows the propensity of an amino acid to take up the helical conformation. Here, the higher the number, the less likely, that means the more energy it would take to stabilize an alpha helix conformation. So it's less likely the higher the number. Here we're showing that example proline as stated, its energy just states greater than 4 because it's so high, proline will not be stabilized in an alpha helix, it will break it. Even other things like cysteine has a pretty high number. So cysteine is less likely to actually form in an alpha helix, and that's because of its R group. Things like arginine are good alpha helix stabilizers, lysine, even, and leucine. So things can be nonpolar, they can be charged. There's various types of R groups that can exist that still help stabilize an alpha helix. But depending on where they are and just generally what type of amino acid they are, can affect the ability of that alpha helix to form. The peptide bonds end up all having a similar orientation in this alpha helix. And again, that is to help with this dipole moment, this positive and negative charge that's occurring based on the carbonyl and amide positioning. So actually as a whole, because of the lining up of these carbonyls and amides along the alpha helix, an actual whole alpha helix has a dipole moment. That means that an alpha helix in total has a general positive end and a general negative end. And again, that's just because all the positive and negative charged groups, or the slightly positive and slightly negative polar groups, the carbonyl and the amide, end up being lined up. So you actually get these, again, polar whole structures of an alpha helix. Okay, so let's now move on to the second of the main secondary structures that can exist within a protein. beta sheets. And again, beta sheets are held together mainly by hydrogen bonding, just like we showed in the alpha helix. Here, a beta sheet is shown where the peptide bond itself is in these flat sheet-like structures. Again, with the side chains sticking out perpendicular to the main chain. To have a beta sheet occur, this backbone of the beta sheet is then interacting with another chain, which I will show. The sheet-like arrangement of the backbone is held together by hydrogen bonds that end up between amides and carbonyls on two different strands. These multi-beta sheet interactions in general, or beta strand interactions, are called sheets. So again, this kind of orientation of the peptide chain we just call a a beta strand. And when more than one of these interact, it is a beta sheet. The main structure, the main secondary structure, is the beta sheet. And again, that is held together by hydrogen bonds. These beta strands, which form sheets, can be oriented in two different ways. And that just depends on the direction of each of the strands next to each other. You can have parallel sheets in which the strands are oriented in the same direction, and then anti-parallel sheets where you have strands oriented in the opposite direction. So here is an example of a beta sheet, a secondary structure beta sheet, and this is a parallel beta sheet. So notice in all of these, if we look along the structure, for example, this amide, and this amide are all lined up pointing the same way. This carbonyl, this carbonyl, this carbonyl, again, all lined up pointing the same way. So these are a parallel strand. They are in the exact direction as the strand underneath of it. These parallel strands can form hydrogen bonds, and that holds them together as this beta sheet. The hydrogen bonding strands, because they run in the same direction, you actually have kind of a little bit more space between the amide and the carbonyl that are bonding with each other, making them a little bit bent and weaker. So parallel beta sheets can exist. Again, they're held together by these hydrogen bonds formed as you go along by the carbonyl and amide groups that are near each other along the strand, but they are a bit weaker when compared to our anti-parallel beta sheets. So here's an anti-parallel beta sheet. You can see, for example, that this beta sheet is in an opposite direction as the beta sheet above it. Here we have a carbonyl of one group and an amide of another group, where we have a carbonyl and I misspoke, excuse me. This is a alpha carbon of one group. This is a amide of one group, and this is a carbonyl of one group. And here we have alpha carbon, carbonyl, amide. So different order. This is carbonyl, amide, carbonyl, amide. This is amide, carbonyl, amide, carbonyl, etc. So just highlighting that they are in the opposite position, opposite to each other. In this anti-parallel formation, the hydrogen bonding between the carbonyls and the amides line up much better. They have a much stronger hydrogen bond, and thus anti-parallel sheets are preferred over the parallel beta sheets. You will notice also, though, that when you have these antiparallel sheets, you have R groups that are pointing towards each other. So here is a carbon, an alpha carbon, a main amino carbon with its R group sticking out. And on the opposite sheet here, we have an R group sticking out as well towards each other. Now on the opposite side here, we have no interaction between R group and R group between these two chains. So every other chain, you have these R group interactions. Now these R group interactions can actually destabilize beta sheets potentially, or they can help stabilize them if they are R groups that interact well together. For example, if they were a negative and a positive charged R group. So just keep that in mind. Every other sheet, you see these R groups that are actually pointed towards each other, which can potentially destabilize or stabilize the beta sheet in the anti-parallel form. Now, what happens when we make these sheets? So you have a amino acid chain like this, and you want to make a beta sheet with another part of the same amino acid chain here. It's making nice hydrogen bonds and forming a beta sheet. Now, these are not just two separate peptides that are existing and making a beta sheet. They're all part of the same amino acid peptide strand. So that means at some point, there has to be a turn that connects one part of the strand to then turn and link to the other part of the strand. These turn areas where it turns to then form the beta sheet. So here's our beta sheets in between these strands. And we have to have multiple turns to allow for the beta sheet formation. These turns occur over a span of 4 amino acids and is also stabilized by hydrogen bonding from the oxygen to amide protons within this turn. Now, one other amino acid that stabilizes these turns is proline when it is in position 2 of the turn. And again, we will show that. Or when glycine is in position 3 of the turn. There are 2 types of main turns that occur within a beta sheet. We call this a type 1 beta turn or a type 2 beta turn. Type 1 beta turn occurs more often, almost twice as frequently as the type 2. The type 2 usually has a glycine in the position of the 3rd amino acid along the turn, if we were to number these 1, 2, 3, 4. And because that glycine is so small, it actually allows for better hydrogen bonding and essentially for more folding and ability of this chain to turn. Now you look, the type 1 beta turn is one that utilizes a proline and it's in position 2. Because proline has that natural R group that actually kinks up a peptide bond. Remember, proline has an R group. If this was our amino acid, here's our proline, and it actually bonds back onto the NH here. So this turn within the chain actually forms kind of like puckering or kinked effect along a peptide, which again is the reason why proline is bad for something like an alpha helix. It will ruin the natural helical formation. But for a turn like this within a beta sheet, proline is perfect. And when it's in the secondary position, it actually helps line up this nice hydrogen bond between adjacent parts of the chain and allows for that turn structure. Now, proline can exist in two potential isomers, and we call it the cis versus trans isomer of proline. The cis versus trans isomer of proline has to do with the essentially bordering carbonyls. You can think of where their positioning is in relation to the proline. And because proline is a fixed structure, you can have the transformation of this bond running through the carbon of the previous amino acid down to the carbonyl off of the actual proline in a trans form. So again, this is where the peptide bond takes on this shape, or a cis form. So here is the shape, the same side versus the opposite side, trans being opposite side, cis being same side. Most peptide bonds that have proline in them are usually actually in this trans configuration of proline. But for peptide bonds that have proline that are in a beta turn, proline is in the cis conformation. The cis conformation, as you can see, naturally makes this turn, this U shape, which helps with forming that beta turn. Proline, again, usually naturally exists in this transformation. And to isomerize it, to actually change it to the cis formation, there is a protein and an enzyme that isomerizes or changes the formation of proline to help with forming these beta sheets. So actual structures of proteins are still reliant on proteins to help them form their structure. It's an interesting kind of chicken and the egg conundrum there. So again, this is how the prolines exist in their state, their isomer state within a beta sheet. Now that's it for secondary structure. We've gone over the two main secondary structures that exist within a protein. Again, primary structure is the sequence of amino acids. Secondary structure exists when those amino acids potentially form either alpha helixes or beta sheets by hydrogen bonding with each other.